The overall goal of this procedure is to perform optical projection tomography-based assessments of the beta cell mass distribution in rodent pancreata, or in tissues harboring islet grafts. Hereby, the near-infrared spectrum is used to enable studies of larger specimens and to allow for the simultaneous visualization of interacting or neighboring cell types.
This is achieved by equipping the OPT scanner with a near-infrared sensitive camera, a high-powered light source, and appropriate filter sets. The specimen is first isolated, fixed and home-mount immunostained before it's embedded in agarose and optically cleared. It is then attached to a custom-made sample holder. A semi-automated positioning tool is utilized to ensure correct centering in the scanner. After scanning, the projection images are post-processed to correct for artifacts introduced by the mechanical setup. If required, an equalization algorithm is utilized to improve detection of low-intensity objects. Finally, segmentation, visualization, and quantification are performed using image processing software.
The insulin-producing beta cells play a pivotal role in maintaining blood glucose homeostasis. Therefore, spatial and quantitative assessments of pancreatic beta cell mass distribution are key to many areas of diabetes research. In this video protocol, we describe an adaptation of optical projection tomography, or OPT, that enables imaging in the near-infrared range and thereby the possibility to image larger bodies of pancreatic tissue. It also enables a greater range of cell types to be studied in a single specimen. We further present a compilation of computational tools developed to optimize OPT for pancreas imaging in general and for assessments of beta cell mass